of this quorum sensing, say, phenomenon, and then engineer them in the shape of positive and negative interactions, and then he materialized these interactions in the shape of different plasmids, and then, well, he put them together, and then what he obtained was a type of bacteria that, depending on the proximity of a signal where he put the autoinducer compound, the cell would turn green or would turn red, okay? So obviously bacteria never do these things by themselves. I mean, this is something that is completely artificial. But in this case, by playing with this quorum sensing and the distance between the original signal that was in the center in the plate and the location of the different bacteria, then at an intermediate concentration of the quorum sensing signal, then the promoter express a green fluorescent protein, but if it had less of that compound, then the promoter the circuit was engineered in such a way that it deliver a red color, okay? Obviously you can play with various configurations. So this paper made it headlines, when it was published first, and it was in the front page of the New York Times, because say, well, this is a case where we engineer bacteria to do something completely different of what they do normally. I mean, you cannot dream of having a natural bacteria doing this type of things, right? But there's something that made still a bigger headline in the New York Times or in the press of that time, was this concept of engineered E. coli strains with light-responsive properties. And this is the work of Chris Voigt. Chris Voigt is one of the big names in synthetic biology today, but by the time he made this work, he was just a graduate student, okay? So there's the possibility of you guys that are starting your careers to really have bright ideas that convert into a kind of embryonic inclination for synthetic biology and for making big contributions to the field. Okay, so how can we make E. coli to develop light-responsive properties? E. coli is in the gut, so in most cases it never sees the light, and well, the idea was the following. So there are a type of microbes that are called cyanobacteria that, as its name indicates, are photosynthetic bacteria. Okay, and how they handle photosynthesis is very complicated, but it happens that they have some proteins that are photoreceptors, and these photoreceptors can be associated to a compound that is called phycocyanobylin, and then they become very sensitive to light, and what happens is that they dimerize when they are exposed to light here. So, and this is an independent domain, okay? There is a transmembrane domain here. Okay, so what he did by that time was to make a hybrid between one of these light receptor domains and an intracellular domain that was phosphorylated when the system was activated, when this protein here could be closer to the other protein there, okay? So in other terms, in the natural context, you have this PCB phytochrome or containing cytochrome that is sensitive to light, but is sensitive to light in cyanobacteria. And then artificially, he fused that to an intracellular transcription sensor that could transfer a phosphorus to a transcriptional activator here. This is called a two-component system. Again, we will discuss that in a subsequent class. But in any case, he engineered a kind of artificial signal transmission pathway in E. coli so that when bacteria were exposed to light, then there was phosphorylation, and then this phosphorus was transferred to a transcriptional factor here that in turn could activate a promoter. And the promoter was engineered in front of a LAC-Z gene. So LAC-Z is one of the most famous reporters in the history of biology. It goes an enzyme that is called beta-lactamase. And you find in the literature many, many color substrates for this enzyme here. And one of the substrates is one that can turn black when the enzyme is expressed. So the kind of concept goes as follows. You have bacteria. Bacteria are engineered with this hybrid transcriptional device. The phosphorus gets here. Then laxate is expressed. And then you have black color in the plates. And he said, OK, so let's do the following experiment. So, obviously, this looks like very messy, right? This is not a high-tech experiment, but again, it's a very intelligent experiment. So he takes an incubator here, and he puts a plate in the incubator that is spread with this bacterium that is engineered in the way I told you, right? And then what he makes is that he puts a picture of himself, a bit kind of chauvinistic, right? And the figure of the photo of his head is projected on the surface of the Petri dish, okay? And the Petri dish is illuminated with a light of the wavelength that is recognized by the photoreceptor, okay? So this is kind of laboratory, setup there. So this is what happened, okay? So this is the picture of himself, of Chris Boyd himself, and this is the image that was projected in the Petri dish. And after a while, this is what you obtain in the Petri dish, okay? So you convert E. coli into a photographic film, right? So obviously, E. coli will never evolve to behave as a photographic film, right? But by doing this trick, you can convert E. coli to do something completely different of what it does naturally. And I tell you, this circuit is super simple. It's not even a circuit. It's just a little device where you convert light into expression of laxate. So this has been, this approach, this paper was the birth of an entire field in biology that is called optogenetics. That is the idea of using light as an inducer of gene expression, or as a way of manipulating, say, gene expression by illuminating different biological, say, bodies with light. Obviously, this is something that was initiated in bacteria, but now it's spread all over the place. You can now illuminate neurons, you can illuminate mice to do this or to that. You can even, there is one gentleman in Switzerland called Martin Fussenegger that has been able to modulate the behavior of mice depending on whether or not they are illuminated with blue light. So, there are some, maybe we will comment as one of the papers I'll show you. Well, this is semi-pornographic, but he has a paper on that. So he has a paper in which he can control erections of the genitals in mice depending on whether they are illuminated or not with blue light, for instance. There's a paper, it's in Nature Biotechnology, it's kind of curious, as I told you, you can propose all type of crazy ideas, and this is one of them. I find them quite funny, that by connecting the liver of some hormones with a promoter that was responsive to light and everything, then he can have mice that are sexually excited or not, depending on whether they see the blue light or not. That's kind of funny thing. Well, funny or not, I don't know. Well, in any case, so this is the initial, the birth of an entire field, optogenetics, and if you put the word optogenetics in Google, you will have like thousands of papers and thousands of comments and so on. Okay, but you say, well, this is at the end a game, right? So you have bacteria producing photographs, producing these nice colors and these kind of patterns and so. But it's just a game, or are we running into something more serious? Okay, so what I have told you so far has like two consequences that, in my opinion, are worth to mention. So one of them is that synthetic biology impacts fundamental science. So synthetic biology is not only about doing things, it's also about understanding things and to having to make sense of biological phenomena and natural phenomena. There is this famous sentence by Richard Feynman. Richard Feynman was a Nobel in physics that is famous for many, many funny quotations, and you can find many of them, you look for them in internet. So he wrote in his backboard this sentence here that has become like a motto of synthetic biology. And is, what I cannot create, I do not understand. So being able to create something is the ultimate proof of understanding, okay? So just keep that in mind, because, synthetic biology is not about doing funny things and light, and no. It's a fundamental, tool in the scientific endeavor, because synthesis is the counterpart of analysis. Analysis and synthesis are the two tools that we scientists use all the time for addressing natural things, right? So, so that means that if you want to understand phenomena, you have to be able to create that phenomena in the laboratory. If you want to understand life, you will never understand what is life unless you are able to create life in the laboratory. It's difficult to understand regulation, make artificial regulatory systems in the laboratory, so on and so on. So creation and engineering is a tool for understanding. As an anecdote, he wrote this in the backboard, and that night he died. So it was the last thing that he put in his life, right? But this has become a big motto in the community. And everyone knows the sentence by Richard Feynman that what I cannot create, I don't understand. But there's this other aspect that is more practical, is more directed to applications, and is in biotechnology. And many of you have this biotechnological interest. And is that synthetic biology enables us to do things better than nature, okay? So sometimes we have this, I think, wrong concept that Mother Nature is wise, and what Mother Nature has created is the best possible solution to every problem, okay? Ladies and gentlemen, in my opinion, that's wrong, okay? And Mother Nature has made a large number of big screw-ups, So, and I think that in many cases, synthetic biology can help us to improve, to do better than nature, maybe in other cases not. But we have the tools now to, instead of depending entirely on evolution, and to use the parts and the components and the activities that are given by nature and evolution, now we can create new things that have never been happened before. So as one of your colleagues mentioned before, for creating means two things, okay? It means to take parts that already exist, but put them in a different configuration. That would be one type of creation. And for instance, as a metaphor, you have the language, So we have words, we have sentences, but the words are always the same. But depending on how we put the words, we can create different sentences or different, say, stories. Or, and then the words and the grammar will be different, but, will be artificial or not, but theory is the same. So we can create new things by combining existing tools, existing items that we combine in a new way, or, and this will be the ultimate screw turn of the whole story, create completely new things. And both things are possible, and both things are in the agenda of synthetic biology. So, what can do better than nature? Well, this slide that is very granular indicates one of the major tendencies in the last few years, nearly a decade, in the world of industrial biotech and industrial chemistry. And it's connected to the crisis of fossil fuels. And I don't need to convince you that we're running out of petroleum, we're running out of fossil fuels. We don't want to burn petroleum or fossil fuels because we release CO2 and so on and so on. So there's this idea that we like it or not, if we want to survive as a human, society, we have to replace oil as the main feedstock for all types of chemical products that we have in our daily life. So where do we find this new feedstock? Well, they may come from different, biological origins that are listed here. So you have starch, you have hemicellulose, you have cellulose, you have lignin, and you have proteins. And the kind of current thinking is that in the future, and even today, we will have, if we are to survive, we have to rely less and less in oil for production of different intermediate compounds, and more in other types of biological masses that capture sunlight and convert them into a material that can be processed to produce different building blocks to eventually having all the products that are the currency of our daily life. So if we ignore oil here, then we can produce with all the other, we can intermediate all these intermediate sugars, or we can produce some syngas, some CO and H2. And then on the basis of that, by using bacterial catalysts, we can generate building blocks here that are converted into secondary chemicals, and then can be converted in intermediates, and then intermediates can be eventually produced converted in different things. So all these arrows here, because we start with a biological material, are amenable to catalysis and to transformations mediated by bacteria that are engineered to that particular end. Because in bacteria or in any microorganisms, you can have a substrate that in many cases are sugars. Microbes like sugars very much, but they have this entire metabolic network that enables you to convert basically any input as a sugar in many, many different types of outputs, just by manipulating the metabolism of different bacteria. And each bacteria or each bacterium has a different metabolic networks with advantages and disadvantages, and with products that are super valuable. We will discuss some of them. But the kind of ambition is being able to get to this list of products and uses to manufacture all these type of products, not on the basis of oil, but on the basis of biological components, because we can produce these intermediate substrates, and then through the action of engineered organisms, being able to convert them into blocks and so on, that at the end translate into different products. So this concept of biomanufacturing is one of the big kind of ambitions of synthetic biology. So biomanufacturing, you will hear along this course like some magic words that immediately attract or capture the imagination of many people. So biomanufacturing, right? So when you think in manufacturing, think in a, in a chain of production with workers there, with robots there and so on. So what if we could use biology to do the whole job or much of the job without having to rely on all these mechanical and mechanical endeavors and mechanical processes, okay? So that will be one of the ambitions. And today, we are not here yet, but I'll give you just one little example in the textile say area. Just as a way of example, no? And if I have time in one other class, I will give you more details. So textiles are typical manufactured things, right? So in the last few years, there's a tremendous interest to replace textiles that are produced through chemical processes by alternatives produced by microorganisms. And in particular, there's this product that is leather. Leather, right? Many of you, some of you have this leather in the shoes or in the belt or wherever. So leather comes from animals. And you know that there's this growing awareness that we should not use animals for various purposes. And so can we find alternatives to leather that don't come from animals but come from bacteria? Okay, so there are various companies. And I have to say that one of the companies, I'm one of the owners, so I'm making a little propaganda here. So that try to convert bacterial biomass into leather or something that looks like leather. Bacteria produce, and I will again talk about that in one of the classes, produce something called biofilms. So biofilms are in 2 dimensions, and there are some bacteria that produce very thick biofilms that can be treated with alkali, and once they are produced with alkali, they produce a material that is indistinguishable from leather, okay? I'm sorry I didn't bring you any example, but you have to take my word to the internet and look for bacterial-based leather, and you will find a number of companies that do that. Okay, so in the stage number one, and this is something that some bacteria do by itself, no need of genetic engineering, synthetic biology, or anything. But think about it, once you have a material that is based on bacteria, not based on animals, you can do so many things with this material. You can functionalize this material. There was a paper just yesterday in which someone called Tom Ellis, that is another big name in the field, made this type of leather with engineered bacteria that produce the bacteria itself a dye of blue color. Okay? So now, by having a biofilm of this bacteria, immediately you have a leather-like thing, a leather textile, that is already blue. You don't need to convert it to stain it with a chemical or anything. Just the product itself will be blue, right? So isn't that amazing, right? That so by making these manipulations, you can really think on biomanufacturing objects and products that until now had to be made by chemical means, and things were very complicated, okay? In my own company, and I'm sorry again, I'm making some propaganda, what we think is a bit different. So instead of having leather functionalized with colors, we think that we can apply some therapeutic value to this compound, so to this leather. For instance, I don't know if in your family, probably you have some patients that are diabetic, And one of the issues in diabetes is to have this diabetic feet that get these sores and that can be infected and it's complicated and it's really nasty. I'm sure that some of you have had some family with that problem. Okay, so what if we could make shoes and have all the characteristics of shoes, but they are based on a live material that will release into the feet environment a medicament that will heal part of the problem. There are known medicaments that can heal part of the problem, but they have to be synthesized chemically or produced in a bioreactor, and then patients have to do that. What if we produce shoes that can be nice, can be fashionable, you can go to parties with them, but at the same time, you add a value to this, would be fantastic, So this is the type of things, the type of application that synthetic biology is after, to look for possibilities and opportunities that don't know thought before, but because of our ability to program DNA, we can now think seriously to do that. I'm sorry for this little parenthesis, but I wanted to share the leather because I think this is a very interesting thing. So once you can program things, once you can program biological objects through DNA, you can have, the only limit is the imagination of what you can do, right? And this is one of the most appealing aspects, at least for me, of synthetic biology. There is no limit to the possibilities that you have in your hands by adopting this type of technologies. What you can do to other things here, but at the end, these arrows in all cases involve having a bacterium that is, or a microbe that is programmed to execute that might be natural or non-natural, but can be programmed. And we'll have an entire class devoted to what is called metabolic engineering. So metabolic engineering is this idea of rechanneling, reshaping the natural metabolic networks of the different microbes to produce something of biotechnological interest, okay? So we will return to that later in the talk. Okay, well, obviously, this type of immense possibilities have not been ignored by what you may call the global think tanks. And in various cases, the World Economic Forum has declared synthetic biology as one of the emerging top technologies for the future of the planet. And well, for instance, in 2012, the World Economic Forum, then they spell out different, different technologies. Number one was informatics. Well, that's very clear. Artificial intelligence, machine learning, and all these things. But then second is synthetic biology and metabolic engineering, right? So like saying, well, if we are to go into another stage of our human industrial societal development, synthetic biology will play a big, big role in it because of these things that I started to kind of share with you, possibilities of doing things completely different or doing the same things with completely different technologies. So I think that's a tremendous thing there. I'll show you just a couple of examples there, and how they are super easy. Detergents. Well, one branch of synthetic biology, as we discussed before, was to create new enzymes that never existed before, but by different technologies, some of them we will discuss, can now be created completely in artificial fashion. So you know that we'll have to go at some time through some laundry. Well, I have to say, I have been in IIT already for 1 week, so one of my immediate, problems is that I have a bunch of laundry that I have to deal in the next few days, but that's a different story. So, but in any case, in laundry involves heating water, right? Because most of the detergents are active only in hot water, okay? So what if we can develop lipases that work in cold water, okay? And this is one of the typical practical endeavors of synthetic biology and metabolic engineering and protein engineering. Well, there are some calculations here of how much energy you could save if you just decrease by 1 degree the temperature of your washing machine, right? And sometimes you have, an immense savings, and you cannot imagine the impact that these little biotechnological advances would have in our general balance of energy consumption. So that would be really, really great. In fact, some of these enzymes have been made and then or improved in some way, and then some calculations have kind of indicated that by having an improved enzyme of this sort, then you can save a lot of petroleum, or if you want to go into biofuels, then you can also save a lot of energy crops. So that's something that, there are people that make this type of calculations. So that's something that is quite curious. Okay, so the last story that I would like to share with you today is something that comes from my own laboratory as an example of the possibilities that you have for using sometimes simpler, sometimes more complicated synthetic biology for many, many applications. So this is a project that was going on in my own laboratory for some time, and I was to show you as an example of the many, many possibilities that you can really deliver with this approach. Bacteria for detection of explosives, okay? Okay, so I don't need to convince you that after wars, there's this big issue of anti-personnel mines that are buried in the soil. So the thing about anti-personnel mines, and I don't know whether you have read something about the problem, is the following. So in military technology, if you want to call it technology, are basically devised to prevent the advance of tanks, okay? And then that means that if you want to a tank, you need a big anti-personnel, big metal mine that explodes and creates havoc in the tank. And this has to be metallic. There's no way they cannot be metallic. So in military technology, and this is, you may consider it evil, but it's the way it is. So and metals can be detected very easily. Metal, there are many ways of detecting metal. Just to avoid that persons go detecting the mines, so these anti-tank mines are put in soil together with these so-called anti-personnel mines. So anti-personnel mines are small explosive devices that are in many cases made with plastic or with ceramics, no metal at all, simply because they are, devised not to be detected. That's the whole point. And therefore, what happens is that after the war is over, and that has happened many times, then you can demine an area by using metal detectors, but there's no way you can detect and remove anti-personnel mines, simply because they don't release anything, right? Okay, so years ago, we came up with the following idea. So in anti-personnel mines, the main explosive is something called TNT because explosive that you have in anti-personnel mines. And TNT, what happens is that it is mixed industrially with another compound that is called 2,4-DNT. You will see the formula in a moment. So this compound is volatile, okay? So how about the following? What if we could engineer a bacterium that goes into the soil, and then the moment that it feels that is exposed to vapors of 2,4-DNT, it emits light. And then this light can be identified by a sensor that moves through the scenario and then kind of pinpoints, careful, you have here an anti-personnel mine. Okay, so this is what is called an area reduction, say, strategy, and it helps tremendously to recover, say, territories or agricultural land that was wasted because of the previous conflict, and it cannot be used because you never know whether one of these anti-personnel mines will explode or not. Okay, so these are the target compounds. This is TNT. This is the explosive, but industrial grade TNT is not pure because of the synthesis process. It's always mixed with some small amounts of 2,4-DNT. This is a descriptor of this of the presence of TNT there. So, what can we do? Okay, what about the following? So we can make a little circuit here where we have a transcriptional factor called SILR5 that is responsive to 2,4-DNT and is based on a precursor transcriptional factor that responds to toluene. I have to give you a little background about that. So some bacteria in the environment are able to degrade this compound that comes from petroleum. It's called M-silane. And then there are different bacteria that are able to feed and to grow on this compound there. And the reason because they do that is because they have a pathway that is called the toll pathway. I will not give you details about that now. That is activated when a transcriptional factor that is present in this bacteria called SILR recognizes this compound here. Now, SILR is responsive to this compound, but by using evolutionary and synthetic biology-related methods, you can make a variant of this protein that, a part of responding to this compound here, is also able to respond to this other compound here. Okay? Now, that means that you can have the target promoter here, that's called PU, combined with a Lux reporter. I don't know whether you are familiar with this reporter. This is a luminescent reporter, that comes from Vibrio. So there are some marine Vibrio that colonizes some organs of fish, and you may have seen these pictures of fluorescent and light-emitting fish in the deep sea and so on. So this is because some of the organs of these fish are colonized by this type of bacteria that naturally light. And obviously this is based because you have genes that do this on a regular basis. So this has become a reporter. So you can take a promoter, put this the luminescent reporter in front of the promoter and have the promoter responsive to SILR5. Okay, this simple circuit, you have here a NOT gate here, you have an OR gate, an AND gate, and an OR gate. Don't worry about the nomenclature because we have some discussion of that in a later thing, but we just make a super little circuit and then you put it in a soil bacterium called Pseudomonas putida and see what happens. So what happens is like this. This is not in soil, this is in a petri dish. So when you have no DNT, then the controls are you have no light emission. This is a DCC camera, so this is a photon counting device. So when you have 2,4-DNT, then in the control nothing happens. Then you see that when you have SILR5, then you have an indication that the reporter is working because it emits light. And then we have another control, transitional regulator, nothing happens here. And then as another control, we have salicylate. That is something that we know activates this other control here. So the basic important device here is this one that you know that reflects. These are bacteria seeded in a petri dish that contain a genetic circuit that implements this logic circuit here, okay? Now, what happens when you do it in natural soil, you know? So in natural soil, what you have here is a microcosm, so it's not a real place with mines, but what we have here is a microcosm with soil, and then buried in this soil, we have either a control or we have the 2,4-DNT, okay? So something similar is exposed to the transitional factor that is not evolved, is not manipulated, is not engineered to respond to 2,4-DNT. In this case, RR5 is engineered to respond to 2,4-DNT, okay? So again, we have 2,4-DNT here, 2,4-DNT there, but in one case, we spray this soil with a factor that has not been engineered, and here a factor that has been engineered. Okay, so what do you obtain? If you use a program for light detection, then what you obtain is that in the two stops, in the two spots where you have 2,4-DNT, you have a considerable light emission, okay? So that means that if you spray this thing in soil, and then you wait for a while, and then you take a photo counting device that surveys the area of interest, then what happens is that you have this signal that will say, here you have an explosive, okay? So this is something that is one of the various cases where you can, I mean, this is just one of the different projects that we have in the laboratory. We have many others, and at given points I will mention here and there as an illustration of different discussions that we have along the course. But you know, it's very clear that you can use these technologies and these logic gates and these kind of tricks to do something that do important things, not only create colors or to make pictures of the PI of the laboratory, but to really do something important. Okay, so we're getting to the end of today's presentations, dear friends, and you know, I want to leave you with this picture here that, in my opinion, is very important, and is the following. So we can chemically synthesize very large molecules of DNA. It's not a big problem today. But there's a big gap between our possibility to synthesize DNA and to engineer, you know, complex behaviors. So at this moment, the best that we can do is to engineer DNA with some kind of reasonable behavior up to 20 kilobases, okay? So that means that you can put a gene, you can have promoter, you can have a terminator, you have another gene, you have this interaction between this transcriptional regulator and the gene here and there, but we hardly can go more than 20 kilobases. But a genome is much more than that. It's maybe in the range of 4 to 8 megabases, and therefore, you know, you are playing not with a few components, with dozens or maybe 100 of components. You are dealing with thousands of components. So we have a gap here between the possibility of synthesizing DNA and the possibility of programming complex behaviors. Okay, and this is in fact one of the big, challenges of synthetic biology that we will try to solve in the next few years with various approaches. And well, whether or not this will be successful, I don't know, and we will see how the fields develop. Okay, there's a kind of a bonus of this story that I told you, and it's the fact that a few years ago we recorded in EMBL in Heidelberg in Germany an entire course on synthetic biology. And you have many talks here, and some of them are really good by some of the names that I mentioned in my presentation. I suppose that you will have access to all my presentations here, so you don't need to take notes of the links and everything. But I don't know whether I can just click here and see at least. Okay, let me see. Okay, just 1 second. Well, I don't know how to do it. Well, anyway. Well, in any case, so just out of curiosity, so there's a course, I think there are like 10 or 12 different talks by different, speakers. I have a couple of them, in case you are interested. I have a short introduction to synthetic biology that is 10 minutes. Many of the things that I say in this introduction is identical to what I have told you already, so you may not be interested. But there are other presentations that are quite interested in case you are kind of curious about them. Okay, and finally, and I don't know what happens here. Let me see. Well, in any case, so the next slide was just the final slide with some reading assignments. So, well, this is not working. Let me see what happens here. Oh, here. Okay. Yeah. Ah, here. Okay. Okay, so you have a whole bunch of different things here that to some extent complement many of the topics that I will discuss here. So you have here my own introduction, then you have synthetic biology circuits, introduction to biology, synthetic cells, gene drives, CO2 fixation, CRISPR, metabolic engineering. This is a very important topic. The other more metabolic engineering. This is a very important one in connection to its societal repercussions or societal, say, spin-offs of synthetic biology by this lady. Then we have building networks. This is the Chris Voigt. This is the guy that made this picture of himself in a petri dish. Now, a few more years later, but he's the one that has developed this amazing tool called Cello for real engineering of complex behaviors through genetic means. Then you have some student projects here. Then you have biosensors. You have transcriptional regulation. Well, you have various things there. So it's really how willing you are to spend some time looking at them, but some of them are complementary to my own talks here. So as a kind of added value, of course, you are welcome to take a look to them. And, okay. Well, okay. And, well, you cannot see what is up there, but these are suggested readings or reading assignments. Okay, so let's try to organize a bit these practical aspects, okay? So, I don't know, what, this is a survey, okay? So, unfortunately today, as I don't have a list of participants in this. I think that you are like 30 people, no, maybe 20. I don't know how many you are, like 20, about 20, right? Okay, so what would you like to handle this practical work? Could you prefer to have teams or would you prefer to have individual assignments? What do you think? Have you? Okay, very good. So we are 20. We can have 4 teams of 5 people or 5 teams of 4 people. What would that make sense? Okay, but the thing is that today I don't have the list of people. So I think we will postpone that until tomorrow because I really need to have the list of participants to have a sensible thing. Okay, so for tomorrow, and I'm sorry that we cannot organize that today because I need the list and all of that. I would encourage you to read the paper that I show you on the repressilator. Please read it in detail, not just the kind of rush overview that I gave you today, and the paper on the switch, on the toggle switch, right? Okay, then what I would ask you would be, well, I will have two papers. It might be a bit difficult. Well, tomorrow will be still a bit messy, okay, because we have to organize this a little more. But as I want you to start presenting your own stories, then I may ask you tomorrow to have a volunteer or two volunteers, one of them to present entirely one of the papers and the other to present entirely the other paper. So I don't know whether we can design the volunteers right now, or you prefer to take a look first to the papers and then decide tomorrow on the spot? I don't know, because if you make a presentation, it would be nice to show some pictures yourselves. I mean, to take the figures of the paper and to discuss them a little as a kind of journal club. I don't know. Should I ask some of you to be volunteer, or you volunteer yourself? Okay, let's try to be gender balanced, So I would like one of the presenters to be a guy and the other presenter to be a lady, So what, I mean, should I say you, or? At the end, everyone will have a chance to say something and to come here and to explain some paper or something. So do not be shy, and should I point someone, Should I? Don't, okay, will I dismiss that you don't want? Okay, so you willing to do it? You have the time?
I have exam tomorrow.
Okay, sure. All of you have exams tomorrow? You too? Is there anyone who does not have exam tomorrow? Okay, you got it. So, okay, you will be one of the speakers then tomorrow?
Okay, what do I have to present?
Okay, you have to get one of the two papers. I will decide now which one. And then please read it in detail, or to the best of your understanding. Get slides of the figures of the paper, and then you come here and make a little summary of the paper and tell us whether you find the paper interesting, what you think is good, what you think, I mean, make a critical assessment of the paper, okay? Because obviously not every paper, I mean, we used to highlight the bright sides of a paper, but obviously every paper has big holes as well. So that's something that we can discuss as well, okay? So it will be, it's okay for you to have this presentation? Okay, very good. And then we need a volunteer, XY volunteer. So who, you?
Yeah.
It's okay? Very good. By the way, I have to confess something that I'm experiencing these days in here, and it's that you guys, when I ask you, do you agree? You say no. I mean, for me, going like this is no. And apparently for you, you move the head in such a way that it means yes, right? Well, I have to learn this body language. I'm unable to do it, but I will learn eventually. Okay, great. Okay, so let's do that. So thank you very much. It will be an improvisation because, we still have to make the groups and everything. And at the end, none of you will escape. I mean, all of you will have to present something at some point, okay? For tomorrow, please, you want the switch or the repressilator?
The
Repressilator? Okay, good. And then you get the other. Is that all right? Okay, very good. So please prepare independently a short presentation on each of these papers, and then we can engage in some conversation about them, okay? And then tomorrow we form the groups and everything, that we organize these assignments with better basis, okay? Okay, so questions, comments? Please, every time that you don't understand me, just let me know, okay? Because I'm very concerned. I'm very heavily accented in Spanish, and so sometimes I'm concerned that I may not be understood, but, as I follow your faces, in most of cases, I kind of feel that you understand me, but one never knows, right? Okay, great. So we are done, I think, for today. Thank you very much. It has been wonderful to talk to you, and I'm sure that we'll have a lot of fun in the next few days and the next few weeks, okay? At least the next couple of weeks. Okay, thank you very much, and see you tomorrow, okay?